Welcome to my channel. Through this video, we will discuss the oogenesis and the vitellogenesis. Oogenesis is a process of maturation of one of ova from the primary germ cells of a germinal epithelium of ovary, and in the female reproductive system, growth process in which the primary egg cell or the ovum becomes a mature ovum, each incorporated. The process of oogenesis is completed in the following step. That is, first is the multiplication phase and the growth phase. Multiplication phase has been subdivided into the following phases, that is growth phase and the maturation phase. So in this, the primordial germ cells of the germinal epithelium lining the ovary undergo the repeated mitotic division to form a large number of diploid daughter, which are called the oogonia. The growth phase include the egg cytoplasm is stuffed with the reserved food material which is needed during the development, so the growth phase is long and elaborative. The growth phase involves an increase of a nuclear and the cytoplasmic substance along with the accumulation of the food reserve, and the growth phase is completed in following phase, that is increase in the nuclear material. The nucleus enter the prophase of the first maturation division, and after the entering of the zygote, the subsequent stages of meiosis are arrested. So, following remarkable events have been reported during this stage: presence of Lambrus chromosome, gene amplification, or the redundancy. So, nucleolus also increases considerably in the case of the amphibian oocyte. Instead of one, a large number of nuclei are formed on the periphery of the germinal vesicle. Next is the growth of cytoplasmic substance. The cytoplasm increases the quantity as well as the quality. During this, the following changes has been reported. The first is the mitochondria, the increase in the number of the mitochondria. Then the ribosome become very numerous and increased protein synthesis required for the formation of the yolk. The endoplasmic reticulum increases and act as a storage site of the RNA in the cytoplasm. Then the cortical granules, the Golgi complexes form small membrane bound spherical body filled with the mucopolysaccharide which accumulate in the peripheral cytoplasm called cortical granules. In yolk platelets, two molecules of the phosphovitin and the associated with each molecule of the lipovitellin. The vitellogenesis, it is a process of yolk accumulation in the ooplasm, and yolk is simply the reserve food in the oocyte consisting of protein, neutral fat, certain carbs, and glycogen, phospholipid, etc. The yolk may be deposited in the ooplasm either in the form of the granules or in the form of the large granules of yolk platelets. And the yolk platelets are formed of two types of yolk, that is phosphovitin and the lipovitellin. It may be synthesized in the body of the female in the liver, and then it is transported and deposited in the egg or elsewhere in the body of female, and finally it is transported and deposited in the ooplasm. The yolk protein are synthesized in the liver, in the vertebrates, on the fat body of insects. The yolk is vegetal pole, interferes with the cytokinesis during the process of cleavage, during to incomplete cleavage. Here is the flowchart of the vitellogenesis. The environment sends the hypothalamus, and the hypothalamus sends the signals to the pituitary gland, which releases the gonadotropic releasing hormones, and then in the follicle cells, the platelet, lipovitellin, and the phosphovitin. This is the part of the oocytes, and then vitellogenin is regulated by the liver function cells. We can easily see that the environmental factors that send the hypothalamus, which sends the positive signals to through the GnRH due to the pituitary gland, and then the ovaries to the liver, and then vitellogenin regulates the ovary as well as the liver. Here is another figure of the process of the vitellogenesis. So it is the pituitary gland, the gonadotropic releasing hormones, the vitellogenesis uptake, estrogen. This is the liver, and this is the part of ovary. So it is believed the mitochondria and the Golgi body are responsible for the converting the soluble form of the yolk into insoluble granules or the platelets. The yolk accumulation in animals and the embryology has isolecithal, maybe the mesolecithal, telolecithal, the centrolecithal. So comes to the another part of, that is differentiation of the oocytes. During the growth phase, when the nutrients and the nucleic acids start accumulating in the ooplasm, gradual differentiation sets in, leading to the establishment of a definite pattern of organization. It is completed in the following step, that is the differentiation of the animal and vegetal pole, establishment of the bilateral symmetry, cortical differentiation, and the maturation phase. The primary oocyte thus form undergoes two successive division. The first maturation division is the reduction division or the meiotic division, which results in the formation of the two haploid daughter cells. And then each haploid daughter cells contain only one member of each homologous pair. Due to the unequal division of the cytoplasm, the cells are unequal. So one with almost no cytoplasm called as the first polar body or the podocytes, and the others enlarged practically with all the cytoplasms called as the secondary oocyte. This secondary oocyte undergoes second maturation division with the unequal division of the cytoplasm forms the small secondary polar body. The first podocytes may or may not divide metatically into the second two, but in each and every case all of them degenerate. Finally, the product of the oogenesis is single large ovum and two or three haploid polar bodies found on the periphery of the ovum. Here is the flow chart of the process of oogenesis. The primordial germ cells enter into gonads, form the oogonium. The diploid oogonia divide repeatedly by mitosis inside ovary called as primary oocytes, and then entry into the meiosis arrest after diploteny of the prophase 1, that is until the female becomes sexually mature. Then the further development of the primary oocytes, formation of the cortical granules, the completion of the meiosis 1, and then the enter into the secondary oocytes arrested at metaphase 2. And in this during this phase the first polar body is formed, and then the completion of meiosis 2 only when the after fertilization, and then the mature egg is formed. Here is a figure of the oogenesis. There is a outer layer known as the theca externa and then in theca interna, both called as the theca cells, then the basement membrane, then granulosa cells, the cumulus oophorus, the corona radiata, zona pellucida, perivitelline space, oocyte, then hillock. So this is about the oogenesis and the vitellogenesis. Thank you.